Hi everyone, I'm Paul Cummins. I'm the instructor for this class on contemporary issues in bioethics, and welcome to the first week one PowerPoint lecture. This week's topic is the incredibly powerful and efficient new gene editing technology known as CRISPR-Cas9. I don't know if any of you had the chance yet to look at some of the extra material in the link under this week's readings. There's an article from the New Yorker that describes the development of CRISPR, and there's also a link to what I thought was an excellent video explaining the CRISPR technology. If you've already looked at that, this slide will be a little redundant, but just to explain to people, CRISPR-Cas9 is a mechanism that combines this CRISPR-associated protein 9. It has the ability to cut the DNA of a DNA strand, while the CRISPR, the clustered regularly interspaced short palindromic repeats, are able to direct which DNA the Cas9 protein will cut using RNA molecules. And so when the cut DNA tries to repair itself, biologists then can change or add DNA at the point of the cut and introduce a alteration to that DNA strand. CRISPR-Cas9 has received a lot of hype. I think if you've been at all tuned into Science or Nature, the two major periodicals within the sciences, but also any of the bioethics journals, you've probably seen that there's been quite a bit of literature produced in a short amount of time on CRISPR-Cas9. And so it seems to be generating even more reaction than previous mechanisms that were developed to edit the genome, zinc finger nucleases and talons. And the scientists seem to think the hype is warranted because of the precision and efficiency of CRISPR-Cas9. And so it's incredibly inexpensive to use. It's apparently an incredibly easy technology to use. And so given the affordability and ease, it will enable scientists to carry out a lot of experiments in a short amount of time and to develop new techniques and new procedures in a short amount of time. And so it seems like it really is a kind of breakthrough technology for genetic engineering that we haven't experienced before. So given the probability that the CRISPR hype will actually be met, and that this is a technology that really will allow scientists to shape the genetics of living organisms in a way that no other technology has. There's a lot of sort of maybe optimistic predictions for the way it could be applied. So I don't really know in industry what the anticipation is, but maybe it's yogurt. Does anybody like Chobani? But so one of the things that I was looking for applications said that if an industry uses bacterial cultures, CRISPR-Cas9 could be engineered to make those more virus resistant. In agriculture, there's predictions that crops will be engineered to resist blight or be more drought resistant. In the lab, scientists can edit single genes in animals to isolate the connection between a gene and its consequences. They can develop much more precise animal models of human disease to study. They can develop potential treatments of genetic diseases by correcting mutant genes. There's the prospect that you could potentially edit animal cell genes to make animal organs safe to transplant to humans. And one thing that I think has probably been in the news with the Zika virus is the prospects of maybe editing mosquito genes to not carry disease. So one early prediction was you could edit mosquitoes to resist the malaria parasite, and then trigger a gene drive to spread that gene through nearly all of the mosquito population, and you'd greatly reduce malaria that way. So I think looking at these, all of these really do seem like promising and positive ways that CRISPR could be used to enhance our lives. Beyond those two applications, those applications on the previous slide, there's also been proposals that CRISPR can be used to edit individual cells' DNA to make them attack human diseases. And so if you were injected with these altered cells, you could fight a disease like cancer with those. And those seem to me maybe further away, but I think in general people aren't too ethically concerned about those kinds of uses of CRISPR. The real use of CRISPR that's caused people to voice special concern is around germline editing in a human embryo. That is altering an embryo's genes in such a way that the alterations are heritable. So the idea is that some embryos will develop genetic diseases because of the genes they've inherited, and CRISPR-Cas9 could be used to edit those genes so they never develop that disease. Of course, once the DNA in the embryo has been changed, when the embryo reproduces, that change as an adult, that change will be passed on to its offspring. And so many commentators have expressed worry about that. However, and this is, I think something that surprised me even when I was thinking about this first topic for this course, because I wouldn't have imagined back in the fall when I proposed this course that by this winter the National Academy of Science and National Academy of Medicine would endorse the use of CRISPR to prevent embryos from acquiring genes that cause serious disease and disability when there's no other way to prevent that. So this is something that is really surprising. The pace of the development of CRISPR is making scientific bodies and ethicists react more quickly and form opinions more quickly about the use of this technology than with the other examples like talons or zinc finger nucleases. So, there's a link to a New York Times article also in that extra readings folder that describes the announcement from the National Academy of Science and the National Academy of Medicine endorsing this. So that is something I just had not anticipated when I designed this class. So in some ways it's kind of a fortuitous event for us to discuss this week. Now, the other surprising thing is that scientists have reported that they've successfully edited the genetic information of viable human embryos. And there had been reports of scientists in China doing this with non-viable embryos with some success, but not great success. But the literature that I looked at on this suggested that the use of CRISPR-Cas9 in viable human embryos was even more successful in editing the genetic information than in non-viable embryos. So now we might see even an acceleration in this kind of research. And so what the scientists did was they gave the embryos a rare genetic disorder. So they engineered them to have a genetic disorder, and then they attempted to cure that disorder with CRISPR. And what they found was that CRISPR completely or partially removed the genetic disorders in most of the embryos that carried them, which is a very promising result for using CRISPR to treat genetic diseases in embryos. Now, it's interesting to note that scientists in China have been at the forefront of pushing the development of CRISPR applications like the one in that article that's also in the extra readings folder. And one of the things we'll see in the readings that you were assigned this week is a worry that if national bodies and ethicists in other countries don't endorse the regulated research in CRISPR in their home countries, that this kind of research will be taking place in countries that have less strict regulatory standards, or that it'll happen, with unscrupulous scientists running it. So I'm not saying the scientists in China are unscrupulous, only that it's interesting that most of these breakthroughs in use of CRISPR on human embryos are coming from China. So in China, they may not have the same regulatory structure that we have. And if we don't, in the United States or in other countries with longer bioethics traditions, engage in regulated ethical research ourselves, we won't be able to have a say in what kind of research should be done. One of the things that we'll see remarked on in some of the articles is that the ethical issues around genetic engineering and gene editing that people raise for CRISPR-Cas9 are not new. We have been thinking about genetic engineering in terms of these issues for as long as science has held out the prospect of this form of intervention into our genes. And so some of the standard kind of ethical concerns people raise are worries about the safety of these technologies. Will they be safe to use? Other issues they bring up are what kind of consequences will come from editing the gene. We can't always predict how the alteration will affect an individual or affect future generations. And without that knowledge, some question whether or not it's ethical to move forward with the technology or even research into the technology. Others argue that these kinds of uses on embryos represent experimentation without consent, and that's not ethical. Others would reply that you can get consent from the parents of the embryos. But as we'll see, a lot of the research that will need to be done will need to be conducted on embryos that are created rather than donated by parents. So it's not clear who would give the consent for that. I think, people express worries about the naturalness of interfering or editing with the historical natural evolution of the genome. Others express a worry that once you introduce treatments, that it's a slippery slope to permitting enhancements through editing the genome. Others, just as they said about assisted reproductive technology, that you're transforming procreation into production. Some people argue that editing the gene closes the open future of an offspring, and others worry about justice. Um, who will be able to access this kind of technology? Will it only be wealthy people? If it is only wealthy people, what does that mean for the rest of us who are unable to afford to use this technology and for our offspring? So these are the ethical issues, and we can discuss them on the discussion board. I'm not going to bring any of them up. I want to focus on the, I'm not going to bring any of them up in great depth. What I want to do is focus on the question of how do we ethically permit research on CRISPR-Cas9.